Hello, my name is Dr. Devendra Lingojwar. I am happy to provide confirmatory test diagnosis demonstration for this kit. This protocol can be divided in 6 major steps. Number 1, sample collection, cell washing, hemolysis preparation. Number 2, keeping cellulose acetate membrane ready for HB electrophoresis. Number 3, keeping electrophoresis system ready for HB electrophoresis. Number 4, hemolyzed application by adsorption method. Number 5, staining. Number 6, destaining. 20 microliter whole blood in 1.5 ml normal saline tube from the EDTA tube in which IV blood is already collected. After collection, mix the tubes in a sample and all sample can be spin for 1 minute. After centrifugation, open. Old sample generally get hemolyzed. Fresh sample today we collected. After washing in normal saline, it will not hemolyzed. So at the bottom of the tube, we will get a packed RBC. So this is called the washed cells. In washed out state, WBCs and immunoglobulins antibody go away with this. So we can decant it and then hemolyze the fresh samples. So just decant this. Blot with tissue paper. So these are the packed RBCs. You can directly add provided with the kit. Some saline is there that need to be removed. Get only packed RBCs without saline. So out of that, just take a 10 microliter packed cells and add 20 microliter distilled water. That is nucleus free water. Hemolyzing solution, that is injection water. Take only 10 microliter of cells in fresh tubes. Fresh tube same level, 1 2 3 1 2 3. So we will take only 10 microliter of red cells. Always use new pipette tip. So every tube contains only 10 microliter of the washed red cells. Add 20 microliter of the injection water. Open this water and store in collection tube. Same water you can use for all the samples. It's labeled as hemolyzing solution. It is nothing but the water, that is injection water. So add double the volume of the washed red cells. So this is called as a hemolysis. Now hemolysis is ready. This we can directly apply for the electrophoresis as a pure hemoglobin protein. Ratio of cells to water should be 1:2. 1 part cells, 2-part hemolyzing solution, that is injection water. Nucleus free water or distilled water also can be used, but it should be double distilled water. If normal saline is not totally washed out, then it will be turbid even after adding water. So with fingertip just tap it. So if you see transparency here in hemolyzed preparation, transparent solution, that means cells are lysed and the pure hemoglobin is now soluble in water. After hemolysis, spin it for brief 1 minute. Hemolyzed is ready. You can keep in fridge or you can directly apply in electrophoresis. Dilution of the reagent. We have provided distilled water to dilute buffer. Electrophoresis buffer 5x. So you make it 1 liter out of this. But if all 100 samples are ready to do, then only for that particular 2 to 3 days use everything. This is 30% methanol for preserving. After opening the cellulose strips supplied, you can use this buffer and use that blue cap rectangular box to preserve the strips. This type of box is also supplied along with that. Then de-staining solution. For de-staining, this is staining and this is de-staining. Staining solution, de-staining. So this is ready to do reagent. This is ready to use reagent. 30% methanol ready to use it. Only this reagent need to be diluted by using distilled water. Distilled water to be used for dilution of the electrophoresis buffer. This is 5x, that means 5 times concentrated. In our laboratory we are using this beaker and measuring cylinder for diluting this. We also provided one graduated tube, that is 50 ml graduated tube, which can be used for measuring the buffer, or in our own laboratory you can use measuring cylinder and one beaker. These are actually very common things available in most of the places. To dilute 5x, we use 50 ml buffer plus 200 ml distilled water. So 5x become 1x. So this 250 ml can add it here. In anode and cathode chamber. ml 5x buffer. So this anode and cathode chambers separated by separator, and these is discontinuous electrophoresis, discontinuous buffer system, and sliders are used here for placing Wattman paper wicks. We have supplied this filters or Wattman paper sheets along in a kit. So this will be used to bridge between the celluloacetate membrane, which will fit here, and the bridge will be from buffer to celluloacetate, then celluloacetate to buffer, another bridge. So 2 weeks we are supplied. We have provided these ready to use papers with double folding. Here we are using single folding, but that is actually better. So just dip this paper in a buffer and keep like this. Now in between the slider, this acrylic slider, and the paper, what this one, the Wattman paper, in between these two, cellular acetate membrane will come. Cellular acetate paper generally we keep it in a refrigerator. These are cellular acetate membrane papers. On this actually we are going to do electrophoresis. This should be preserved in 30% methanol. So in kit we have provided these papers in packed condition, horizontal and straight, and also empty box is also provided to store this. So first we have to wash out methanol, whatever 30% methanol is there, we need to wash it out in tap water. Acetate membrane paper and wash it out means all the alcohol should go away. So wash for 1 minute. Take care not to fold the paper. After washing there is no oiliness on the paper. So 2 to 3 times you can change the water and continue for 1 minute. Wash it out. It will look like without oily patches. There is no oily patches in this. Now we have to wash it with also distilled water is also given there. So if required, we can give a 1 wash of distilled water also. So tap water traces will not be there. This with the buffer. It is better to dilute buffer in a beaker. If it is not there, you can have the one beaker, and the same buffer you can use it for soaking also. So I will prepare again 1x buffer. So for 1x we require for 100 ml 20 ml plus 80 ml. 100 ml is sufficient for soaking. So soak this paper, cellular acetate membrane paper, for 10 minutes. So total 20 minutes we can keep for soaking in buffer. So change from first change to second change for soaking buffer. Keep it for 10 minutes. In between experiment, you can use this tissue paper for blotting this paper and labeling. Here right hand cut is given, so this right hand cut should be used for applying the samples. So you can keep this way and just blot it. Just keep half centimeter from here and use it for labeling. Wherever white part is there, we can use for adding markings. 16, 17. So along with control, 17 line can be drawn. These two can be a control, first and last. So in between you can soak in a electrophoresis buffer. So at any time point it should not get dry. Since we have only 3 fresh samples for the demonstration, we will not use the entire strip, but this we have given demo for how to apply for all 15 samples in one strip. So for the running 3 samples, we use it the small strips. So we can repeat for once more. So we can have 6 together. Again give the same cut to identify. Use tissue paper to blot once again. I'll write in here the sample number 123 123. As per the number you can change that, or you can have the control like AS is control, SS is control. Here just we are adding only numbers. Carefully place this cellulose membrane in between the slider and the Whatman paper or the blotting paper. Moist it with the buffer, and as per the sequence you can start sample application, then the electrophoresis. You have to apply the sample now. Before that you can pre-run sample should be applied to the negative electrode always. In the that particular piece we have given to you, negative electrode is coming here and positive electrode is making you in this direction. So you can apply this strip the other way around. So in any way there should be negative sign here, and there should be the sample application should be towards negative A. 100 volt for 30 minutes is sufficient for sample separation between S and A hemoglobin. So everything is correct now. This is very basic model of electrophoresis. For cellulose acetate membrane electrophoresis, if we apply 200 fixed volt, we will get 8 milliampere output current. So that is working fine. Buffer is crystallized. Keep the cellulose strips. Now we'll keep the cellulose strip there and check current. Now this is the continuity, buffer, brick, and then the cellulose gel, then again wicks, and then the buffer. Now this is continuity. So once you start enter, it will show current on and 5 milliampere, 4 milliampere. So instead of 0 it is showing, that means current is flowing. So this in simple term it is already given in a supplied instrumentation with the 2 output and showing current as well as voltage there. And this one is very basic. This one is very basic. Sample. We need to have distilled water also, just to wash in between two samples. So the same distilled water we can use it. Applicator is given in this type of tube, in 15 ml tube. Applicator is given, and these are the samples. Same tissue paper, we can use it for blotting. Number 1 sample, just do not press it, just go inside the tube and touch. You get this much sample, and that is sufficient for loading. Apply at one place. So we will turn it off. Just press and apply. I will apply sample number 1. Here you can wash out. Just press and release the pressure. It will wash out. I am applying duplicate the same sample. Wash every time. Since it's a duplicate sample, same sample down. Wash here. Just press and release, press and release. See the movement of the hand. Just apply the sample very delicately. Press and release the thumb. These are all normal blood sample taken in lab just for the demonstration. Actual sample if we get, once again we'll send you with AS SS controls. Now just start this machine. Press. It will show current flowing 5 to 4 milliampere. The bands will migrate from negative electrode cathode to positive electrode anode. Proteins carry negative charge, so these are anions. Anions attracted towards anode. And apply always towards negative electrode. Same like DNA electrophoresis, we apply towards negative electrode. DNA carry negative charge, it will go to the positive charge electrode, that is cathode anode. Same for the protein at 8.6 pH, alkaline pH. The protein carries negative charges and it goes towards positive electrode. Now bands are now migrating. We have given dropper also 3 ml. It is just for soaking or just keeping this membrane wet condition. Generally in summer conditions the strip get dry during electrophoresis. I wait for 20 minutes. Since these are only for the demonstration purpose normal hemoglobin, it will not separate into A and S, but when solubility test positive samples you apply, those having the sickle cell carrier, they will split into two. I will show you here. Two bands AS, single fast migrating band SS, two bands fast and slow AS, and slow migrating band SS, and these two are controlled AS. More precisely you can see in electrophoresis already done for previous samples. These are actually done electrophoresis from field work. So when you go to the field where sickle cell is very prevalent, we can see such results. AS control, SS, AS, then this is AA, then AS, then A, this is SS, then AS, 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 then AA, then SS. So wherever solubility tests are positive, more and more sample will see positive samples, either SS or AS there. Ideally, you should get this type of separation, two clear bands separated. So within half an hour, we can see two clear bands get separated. So these all are AS samples. Only one is AA. SS is not here. Since this is unused strip, we can just wash this and keep it in 30% methanol for the next use. And we can wash it with distilled water or tap water. We will be keeping this in 30% methanol for next use. After 20 minutes, mostly 20 to 25 minutes, before this band touch to the Whatman paper brick, you can turn it off. Carefully remove the cord and remove the strip. Place in a box given for this staining. You can add entire 100 ml stain and keep it for 1 minute. Shake vigorously for 1 minute. After 1 minute of shaking, collect stain in the same staining box and keep it for the next use. And excess stain need to be removed from. De-staining. This is ready to use de-stainer. So use de-stain it with vigorous shaking until the background get totally white. At the end of the de-staining, within 1 hour, you will get a clear result. You can see here all are normal. So all samples run towards anode and all are normal, A plus A. If you compare with this, all are A plus A sample, that is normal adult. Here if it is carrier or patient, then presence of S should have been seen here, 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 here. So in SS, the band will come here near the point of application just with half centimeter migration within 20 minutes, and that will be SS. So both the patterns S plus S is sickle cell disease. If fast migrating band like this is A plus A pattern. If there are two bands A and S, one slow migrating and one fast, then that is carrier, that is AS. So that way we can confirm sickle cell anemia. So this is just for the demonstration purpose. If we get control samples, we will give a demo along with the control sample also. But whenever we have the solubility test positivity in your area, so most of the samples you get AS and few will get ASS. This is a small strip with pattern of AA, AS and SS. This right hand side we have today's work, and this is earlier work where we have known samples of AS, AA and SS. So first is AS carrier, 2 bands, then AA single band fast migrating, then AS 2 bands slow and fast, then SS slow migrating single band, then AS 2 band, then AA single band then AS 2 band. So this way we can diagnose sickle cell anemia wherever SS is there, single slow migrating band. Nanded region where we got most of the samples only AA pattern. So AA pattern will look like this only. Although we applied control AS there, we got all normal AA only. And this is the case where we applied, we got only one carrier there, 59 number from Nanded. SS is a control from earlier studies, and rest of them were only AA pattern. So by using this single slow migrating SS, then one slow migrating, one fast migrating 59 number AS, and rest of them are only fast migrating single band. All are AA pattern. So this way we can diagnose sickle cell anemia. After electrophoresis and your experiment is over, you can keep things as it is. Just cap this micro applicator, decant this electrophoresis buffer. This also can be kept in a refrigerator. So we can use this papers also the next day. And this buffer also should be kept in. So once you prepare buffer, at least for a week you can use it continuously. So whatever we have supplied for 100 tests, it will go for more than 200 tests actually. This is stock buffer. Working, you can keep it in the beaker, and then you can keep it in fridge. Wash the tank in water, tap water, and keep it inverted like this, inverted position to get a dry. For long term use and long term stability for electrode, every time you use this tank, at the end of the day you should use it clean it properly, and then you can use next time as a dry conditions. So always keep it in dry conditions. This is the way we can do confirmatory testing of sickle cell anemia by using cellulose acetate membrane electrophoresis at alkaline pH. Once lab technician get enough training and practice, they can easily do at least minimum 15 samples in a 1 hour initially with good practice. Most delicate thing is here how to apply samples by using the applicator by adsorption method. Rest of the things are very common in most of the laboratory. Now interpretation about the sickle cell anemia. Once you apply sample to the cathode end, within half an hour sample migrates towards anode end, and at the end of the electrophoresis you get 3 type of band pattern. Normal hemoglobin is A plus A, that is it will be fast migrating band. Sickle cell carrier where you get a 2 bands, one A and one S. And sickle cell anemia disease patient where you get a slow migrating single band. So based on these 3 types of band pattern migration, sickle cell anemia can be confirmed. That is normal adult A plus A, sickle cell carrier A plus S, and sickle cell disease SCD, that is S plus S. Thank you.